Recording. That's me. Adjust that a little bit. Wish I had a. Oh, you know what? If I put a little blinder on there, it might make glare. I think rid of that glare. Just gonna have a glare. I want the lighting in here. Okay, let me share my screen here. Let everybody in. This is section 90, right? Let's see. Let me get that out of here. Where's my? Not this. I need Histology Slides. There it is. Ta-da. I'm going to change my display. There we go. All right, we still have 3 minutes, 2 minutes actually. Letting in some stragglers here. I guess you're not a straggler if you're not late yet. So, got 14 people in the meeting right now. That includes me, so 13 students. Well, here's another person, and another, and 2 more. Well, they're starting to file in now. Get a fresh water. Oh. Put that right there. Oof. Oh, look at you guys. Yeah, nothing I can do about that. Just gonna have this annoying glare on my image. Just letting folks in. Okay, almost ready to start. There we go. Now it's 10 o'clock. Today's Wednesday, January 27, 2021. And it's 10 o'clock. You guys are my biology 1161 section 90. Wednesday, 10 o'clock section. And remember, we're into histology. That's what we've been studying, the histology slides. Remember, it's 13 slides we're looking at. Last week, we looked at like the first 4 or 5 or something like that. And so we're just kind of heading along, picking up where we left off. Give me my laser pointer here. We looked at the integument, remember that? We looked at the esophagus, remember that? And we looked at the kidney, remember that? And then the ear, the pinna, the external ear, or the little mouse. And here we are, this is where we left off. So today, this is our first slide, the epiglottis. First of all, you know what the epiglottis is? You remember what the epiglottis is? It's that flap that closes over the opening to your voice box when you swallow. The opening to the larynx is called the glottis. Get it? Glottis, epiglottis, above the glottis, it closes over. And here it is. This is a section taken. It's not human. Probably taken from a little mouse. So we put the whole thing on a slide here. This is magnified maybe 100 times. And if you think about it, it's constructed a lot like the ear. Remember the pinna. This is why I show it to you right after the ear, because the pinna is skin on either side of elastic cartilage, epithelial tissue on either side of elastic cartilage, and that's exactly what you got here. Is you have elastic cartilage in the middle, and then epithelial tissue on either side, and of course underlying connective tissue and so on and so forth. But of course, this is inside your body. It's not a stratified squamous that's keratinized. You're never gonna have keratinized epithelial tissue inside your body. And if you look like at your list of tissues, you'll see, and this first slide tells you right here, the superior surface is stratified squamous epithelium, the inferior surface, the respiratory epithelium. That would be pseudostratified ciliate columnar epithelium. So you go, what? Yeah, that's right. You have one type of epithelial tissue on one surface, and then it segues and it turns to another type of epithelial tissue on the other surface. This is the inferior surface. That's the surface that faces the glottis, the opening, when of course it's going to be continuous with the respiratory passageway. And you'll see when we look at the lung and the trachea and so forth, it's the same type of epithelium, the respiratory epithelium, which is almost always pseudostratified ciliated columnar epithelium, and we usually abbreviate that PCC. So you got PCC here and stratified squamous epithelium here, but non-keratinized, right? And so why is that? Well, this is the surface that is closing over the glottis, and that's what the surface that food and whatever else would, scrape against. And so this epithelium is continuous with the epithelium that's in the back of your throat, in your oral cavity, your mouth. The entire oral cavity is lined with non-keratinized stratified squamous, as well as your pharynx, and then also your esophagus, we're going to see. So this is all the same stuff, but it changes. It segues to a simple epithelium, because you don't need that extra thickness on the layer that's not exposed to an abrasive trauma. And so that's why that's the way it is. Okay. It points out a couple other things here for you. This is like a new term, I think. I don't think we've had this term yet. Lamina propria. Lamina propria. That's histology buzzword. That refers to the associated connective tissue of an epithelial tissue. So it's the underlying epithelial tissue. Now, all epithelial tissue has a lamina propria. It's kind of like the tissue that's underneath the basement membrane, but it's, like a continuation of the basement membrane, the lamina propria. Well, in certain areas of the body, the lamina propria has a special name, and you know what this is. You've already had it. In the skin, your integument, there's an epithelial tissue, remember, keratinized, stratified squamous. The lamina propria of the skin is the dermis of the skin, okay? The lamina propria of a intestinal mucosa, or what did we look at? We haven't looked at that yet. Or some kind of like a mucous membrane. If you have a mucous membrane, the lamina propria of a mucous membrane would be the submucosa, okay? And so that's what it is. It's just the underlying connective tissue. Now here is the skeletal element. Now, a lot of times when you think skeletal, you think bones. You say, that's a skeleton. Well, no, it's cartilage. But what a skeleton is, by definition, it provides structural support inside. So it gives it a form. Like your ear, remember your pinna or your auricle. Humans have an auricle, they don't have a pinna. It has a shape. You know, it has to have some kind of structural component inside of it to give it its shape, and that's the cartilage, the elastic cartilage. Well, your epiglottis, same thing. It has a distinct shape, and it's made out of the same kind of cartilage. And by the way, this is the only two parts of your body you're going to have elastic cartilage. The ear, external ear, and the epiglottis, the only two places. And so if you know that you're looking at the ear, or if you know you're looking at the epiglottis, you should expect elastic cartilage. And remember, elastic cartilage is one of three different types of cartilages that you have in your body. And what makes elastic cartilage is it possesses the protein fiber elastin, elastic fibers. And you'll see elastic fibers in the matrix once you magnify them. If this magnification right here, you can't see it. You just kind of tell, oh, that kind of looks like cartilage. But next slide should show a little bit closer view. And so we'll look at some of the tissues in there. So here, this view, we're just looking at the overlying stratified squamous epithelium that's non-keratinized. This is the superior surface. And you can tell, in fact, that, gosh, that's definitely stratified. You can see several cell layers thick. And it's squamous cells. You look at the apical cells, they're flat. So stratified squamous, all right? Stratified squamous. Now, we saw stratified squamous, remember, on the skin, but it didn't look like this. This is doesn't have that dry flaky layer, so you know it's non-keratinized, okay? This is the other surface, the inferior surface, where you have the PCC, pseudostratified ciliated columnar epithelium. Get it? PCC. Here it is. This is columnar cells, but then you have this underlying basal cells, all right? And that's stratified squamous. Every once in a while, you might see a goblet cell. And ciliated, it has cilia. This particular view, I don't see a lot of, they're there. It's just kind of hard to make out. We'll have to get a different specimen to see some really good cilia. And we will. I'm going to show you some good cilia, but this is what it looks like here on the epiglottis, on this particular specimen anyway. All right, PCC. This slide right here shows you some of the connective tissue. Beginning, well, let's look down here. This is the, I guess, the most important one in the center. That's the elastic cartilage. And you can tell, first of all, it's cartilage. All cartilage has one characteristic that's just hand down cartilage. All cartilage has these pits. You see these little holes? They're called lacunae, L-U-C-U-N-A-E. That's plural for the word lacuna. And I can't remember if I told you about this before or not. If I did, just, I'm sorry for being repetitive, but a lacuna means little lake, all right? And what cartilage has these little lakes, they're the little ponds, little lakes of interstitial fluid to accommodate a cartilage cell. So inside lacunae, you're gonna have chondrocytes. A chondrocyte is a cartilage cell, because the matrix of the cartilage is firm, it's solid. You know, like you've seen cartilage before, gristle. Have you ever had gristle from a chicken bone or something like that? Not the cartilage, or not the bone, but the cartilage, it's the gristle. And it's not hard and mineralized like bone, but it is stiff and firm, but it's slightly pliable, okay? But it's too dense for, material to diffuse through it, and the living cells need a space to hang out and live. And so they all live inside these little pits called lacunae. And so we'll zoom in on that and look, but this is all definitely cartilage. But of the three different types of cartilage, this is the only one that kind of looks dirty like this. See, to me, it kind of looks like dirty hyaline cartilage. And what gives it that dark, dirty look is the presence of the elastic fibers. And so the elastic fibers gives it that very dark look. If you look carefully, you can see some little bands, little squiggly lines. Those are the elastic fibers that you see in there. So it's only going to look like this in two places, the ear and the epiglottis, elastic cartilage, okay? Now, you look at the edge of the cartilage right here. See this little bracket right here? Dense irregular connective tissue, the perichondrium, all right? All cartilage, it doesn't matter where it is or what type it is. It could be fiber cartilage, elastic cartilage, or elastic fiber, hyaline, elastic, and fibro, okay? Doesn't matter what kind it is. All cartilages are enveloped in a fibrous connective tissue membrane. And that's what it is right here. You can see that these are collagen fibers wrapping the outside of the cartilage. We have a name for that. The membrane that surrounds the outside of cartilage is called perichondrium. Peri, surrounding, chondrium means cartilage. So the perichondrium is always made out of the same stuff the reticular layer of the skin is made out of, and that's what it looks like. It looks just like that, but that's not what it's doing. You know, this is the dense irregular connective tissue forming the perichondrium associated with the cartilage. Okay, and it basically this fibrous connective tissue envelope, essentially on the outside of cartilage, is there as a substrate for things to attach to, and so it's going to allow the superficial tissues on that to anchor to it. Okay, so there you go. And then this next bracket right here, this would be more superficial, going out, you know, towards the top or the outside, I guess. And you can barely see, this is the fat layer, so adipose connective tissue. It kind of looks like little rings. There's a little adipocytes, little fat cells. You see them everywhere. You see a few fat cells over here. You have fat anywhere, but this is a very distinct layer right here. You can tell it's something different. This is different than this, and this is different than this, and so on and so forth. So this is a good slide. You know, I would probably use this one on a test. You know, I point to this tissue, say, name this tissue. And you should recognize it as elastic cartilage. So name this tissue right here, and you should recognize this stuff right here as dense irregular connective tissue. And of course, this, I can say, hey, name this tissue right here, and you should recognize that as adipose connective tissue. Can you handle that, you think? I bet you can. It's not hard. More of the epiglottis. Here, this is a better shot. I'm thinking, well, I don't know if it's better. It's different. How about that? It's the epiglottis, but it's showing you the epithelial tissue right here on this bracket. It might be kind of a challenge to you to say, what? That doesn't look like textbook example of pseudostratified ciliate columnar epithelium, PCC. It doesn't really, but it looks terrible. Look at that. But sometimes you gotta think. You know, you gotta use your brain and figure it out based on, you know, what it could be. You know, now if you know that you're looking at the epiglottis and you're looking at this, you should know hands down that this has got to be epithelial tissue because it's on the outside, right? Well, what kind of epithelial tissue could it be? Well, there's only two types it could be: non-keratinized stratified squamous epithelium or PCC. Think about that. PCC, that's the columnar versus squamous. Why am I doing my hands like this? Columnar cells have a vertical orientation. Squamous cells have a horizontal orientation. You tell me, does this look more vertical or more horizontal? It looks more vertical. So I would call this columnar. Yeah, the pseudostratified ciliate columnar epithelium. And then here's some underlying connective tissue. This stuff right here, I didn't. Oh, well, I did. I didn't label this. I did it over here. This is areolar connective tissue. Remember I said areolar? It's kind of like a space filler. Remember we saw areolar in the papillary layer of the dermis, and it's kind of the same zone, same area. You don't want it to be dense connective tissue in there because you're going to have blood vessels and glands, and these are some capillaries right here, and so you got to make room for that. To make room for those kind of structures, you need areolar, not a dense connective tissue. Okay, and that's it. That's the epiglottis. Okay. Next is the lung. But you know what? I gotta look at something real quick. Let me do something. I'm going to show y'all. Did I show you? I can't remember if I showed this class your checklist of tissues. Let me open that real quick. I thought I had it open. I guess I don't. Let's see. This is from my courses, and it's what I used to give my lab students before I built this PowerPoint. And the PowerPoint's based on this. I wonder if this is it. I think this is it. Let me see. So, yep, this is it right here. This is from whatchamacallit, my courses, and it's underneath, I guess, material for practical 1 or something like that. And it's just a list of your 13, remember your lucky 13, and then order, and it lists all the tissues and so forth. And here's the pinna, the epiglottis, and then the lung. And let's see, epiglottis, that's 5. 6 is the lung. 7 is, okay. Yeah. Okay. And so what do we, I'm trying to think where we stopped today. I think the xiphoid. I can't remember. Or you know where I could look? It's on the course policy sheet. Let me look at that. Let me do this. Let me open. That's, where is it? There it is. Right. Okay, yeah, xiphoid. Okay, I'm trying to figure out where we're stopping. Okay, so here we are at the lung. And there we are. You like the lung? I like the lung. Because, I don't know, it's just got some really great tissues in it. And let me get my pointer back. Nothing looks quite like the lung. There's a section of the lung magnified about 50 times, 40, 50 times, something like that. And your field of view, when you're looking, you just see this stuff everywhere. Nothing else looks quite like this. What is all these little spaces you see everywhere? Those are all the air sacs in the lungs. Collectively, the alveoli. One of them would be an alveolus, but bunches of them in the alveoli. So they're everywhere. And then every once in a while, you see implanted amongst all those alveoli, a blood vessel or a bronchiole, okay? So these are the structures you're gonna be looking at: alveoli, blood vessels, bronchioles. And of course, each of those are gonna be, made out some kind of tissue and so forth. So what are they? Well, here we are zoomed in about 100 times. Again, alveoli everywhere, bronchiole, blood vessel. Okay, just to get your bearings, what you're looking at. So what are these tissues in here? Well, you zoom in, I kind of show you kind of like this. This is magnified about 400 or 500 times inside a bronchiole. And this is a beautiful, I love this specimen. Look at it. Pseudostratified clump. This is your PCC textbook. Just look how beautiful and detailed these cilia are up there. You cannot confuse this with anything. You got your columnar cells, your basal cells, and look, I'm looking for a goblet cell. I think that's a goblet cell right there. It's kind of grainy looking. Maybe another piece of a goblet cell. Yeah, and then here's one right here. Sometimes they're clear. If you look and you just kind of see just an oddball looking cell it's making its connection to the surface and not ciliated, that's a goblet cell. So ciliate columnar cells, goblet cells, basal cells. Yeah, I got them all labeled right here. That's PCC. That's lining the inside of the bronchiole. And a good specimen of the epiglottis, this is what the PCC should look like on the epiglottis, but I found this in the lung. How about that? Isn't that nice? Here's a close-up of alveoli and the endothelium. Remember the alveolus, the air sacs of the lung, it's thin, thin, thin. It can't be any thinner than 1 cell layer thick. And so that's simple squamous epithelium. And then lining inside of all blood vessels is also simple squamous epithelium, and it's called endothelium. Endothelium is just the inner epithelial lining of a blood vessel. And it's always, always, always simple squamous epithelium. So if you're looking at it, and you kind of see it, this little flat little cells lining the inside, flat little cells right here, simple squamous. These are alveoli everywhere. And it's, you know, look how thin. It has to be thin like that to allow for respiratory gases to go through it, right? And of course, all blood vessels, you know, start arteries segue to arterioles, arterioles segue to capillaries, capillaries segue to venules, venules to veins, etc. But the capillaries are just like the alveoli, thin, thin, 1 cell layer thick. And so that simple squamous of the capillary is the same simple squamous that's inside of all your blood vessels. And it's continuous with the inside lining of the heart too. But in the heart, if you're in my lecture, you hopefully remember this. Inside the heart, we don't call it endothelium, although your book does. I don't. You gotta do what I say, and I don't care what the book says. But inside the heart, the endothelium, we call it endocardium. So endocardium, endothelium, the alveoli, it's all simple squamous. There you go. Now, inside the lung, of course, you're going to find pieces of cartilage. And the cartilage supplies the skeletal element for the bronchioles, okay, the internal structural support holding the bronchioles open, okay? But it's not elastic cartilage. And there's 3 different types of cartilage: elastic, hyaline, and fibrocartilage. Hyaline cartilage is probably the most common one in your body, most diverse. It's everywhere. It forms the articular cartilage of your joints, most of your joints anyway. And that's the cartilage in your nose, the cartilage in your ribs, etc. And it also forms tracheal rings in your trachea, and then descending down from the trachea, the bronchi, you got it. And it's a beautiful specimen of hyaline cartilage. And it's just like elastic cartilage minus all those dirty fibers. The word hyaline means glass-like, like a pretty stained glass window in a church. And it's because the tissue is pretty translucent. You know, light passes through it pretty easily. Elastic cartilage looks like a dirty stained glass window. It's like an old church that's been abandoned. It's got spider webs all over it, and the spider webs would be like the elastic fibers. But here you got a nice, pretty, clean stained glass window, minus the little pits in it. And you see the little pits and the dark staining structures inside the pits, or the actual cartilage cells. So those are the chondrocytes and lacunae. Same thing, all cartilage has chondrocytes and lacunae, but only hyaline cartilage is fiber-free. There's no fibers in it whatsoever. It's just clear, you know, hyaline cartilage is translucent matrix. So there you go. That's a nice example of it. Here's some of your PCC up there, your lamina propria, the underlying connective tissue, a little band of smooth muscle. There's some great tissues here. Good stuff. All right. I think the main thing I'm trying to point out here is the hyaline cartilage. All right. More lung. Oh, here I'm showing you smooth muscle. Again, smooth muscle adjacent to the bronchioles. And then also, you're going to find smooth muscle in the walls of little arteries, okay? And you can see these smooth muscle cells kind of going around. That's what gives the bulk of the wall its thickness, that smooth muscle, okay? Look, there's that word endothelium, remember? Endothelium, that's the simple squamous epithelial cells that form the inner lining. That's good, okay? So that's a good picture. And again, remember, still a lung slide. Beautiful picture right here of the hyaline cartilage that's, in there supporting the bronchial. Here's the inside of a bronchial. But remember I said all cartilage, doesn't matter what kind it is, it's always going to be wrapped in perichondrium. So you can see right there at the edge, this is a different tissue. It's thin, but it's not cartilage. Right there at the edge, you see that there? And then right here, that's dense irregular connective tissue forming the perichondrium. So you always have perichondrium there. This is, I don't know if you recognize this up here. I mean, there's all kinds of, there's tissue everywhere. You should recognize this stuff. I don't, maybe I pointed out, but that's it. That's smooth muscle up there. I didn't point it out, but there it is. But I think I already pointed it out somewhere else. But this slide right here, this is pointing out blood. Now, blood is a connective tissue. It's known as vascular connective tissue. And so I'm just pointing out, hey, this connective tissue in the blood vessels. Here's a little artery, arteriole. Here's another little artery. This is smooth muscle in the wall. But the little cells you see in there, and we're going to look at the types of cells and so forth when we look at just a blood smear. But for right now, if I pointed in here and I said, name this tissue or name this connective tissue, I mean, you should call it vascular connective tissue, which you commonly refer to as blood. Okay, so there's that. And that's it. That's the lung. Okay. Next, we're going to the trachea. Now, look at this. This is a great slide. This is more than just the trachea, but I wanted to include it so you can kind of get your bearings. This is a horizontal section, through the throat, and so you're traversing the trachea and the esophagus. This is anterior, this is posterior, and then the spinal column would be like right here, right? So your esophagus is always going to be deep to your trachea. Like if you have a tracheotomy, it's in the front, right? And behind your trachea is your esophagus. I just kind of want to show you this. And of course, both of these organs are hollow organs, and this would be the lumen here and the lumen here. But notice how nice and round the lumen of the trachea is versus the lumen of the esophagus. Remember, the esophagus kind of collapses because this normally stays closed because you don't want like food and stuff spilling out. I mean, you can stand on your head to make you puke, right? Because it's always closed. But your trachea always is open because it has these cartilaginous rings that are called tracheal rings, about 16 of them in the human trachea that hold it open all the time. Because that's your windpipe. It's the same reason you have rings on your vacuum cleaner hose. You ever notice that? You have like a garden hose and you have a vacuum cleaner hose. What's the difference? Well, garden hose is just kind of rubbery, whatever. It's pressured, from the inside out. Vacuum cleaner hose is a negative pressure. It wants to collapse, but to prevent collapsing, to keep it open, it's equipped with these rings. You ever notice that? That's to hold them open. And so your trachea is like a vacuum cleaner hose. It's supported with those little tracheal rings. And so here we're going to zoom in on that and looking at it up close. Again, the inner lining, that's the respiratory epithelium, right? Which is pseudostratified ciliary columnar epithelium, always. Now this is only magnified maybe 50 times, 40, 50 times. So you can't really see the detail, barely see some little cilia, that little fuzzy little edge, that's cilia. And if you see cilia, at least in my class, it's always going to be pseudostratified. It is true that there are other epithelial tissues that can be ciliated. The only other kind that it could be would be just regular ciliated columnar, but we're not looking at that. So in my class, the only thing that's going to be ciliated is pseudostratified. And it is also true that pseudostratified does not have to be ciliated. But again, in my class, you won't see non-ciliated pseudostratified. There's only two parts in your body that you can see that, the male urethra and the eustachian tubes. We don't look at either of those two, so you don't have to worry about that. So the bottom line is, if you see cilia, you're looking at PCC. And if you're looking at PCC, it's in the respiratory system somewhere. So this is the trachea. You get your little band of PCC right there. Here, of course, is the cartilage that's holding it open. And it's hyaline cartilage. See, it's nice and pretty and clear. You can see lacunae with chondrocytes, the whole nine yards. And this is a cool little patch of fat, adipose connective tissue. See the adipocytes there? And then look at this. I didn't point it out, but where do you stop? You got to stop somewhere. On this slide, I guess I just stopped at these three. But if you wanted to see, You know, recognize what that is? That's a little blood vessel, and that's blood. You know, you would have endothelium here, which would be simple squamous, and then this stuff right here, that's vascular connective tissue. That's blood. Okay? Yeah, I mean, you can have stuff everywhere, but I just kind of picked out things that I wanted you to see. And what you need to know are the stuff that I pointed out with the arrows. Okay, and that's what I'm going to ask you on your test. What am I doing here? Why did I do that? I think that was probably just a boo-boo on my part. It's pointing at the same stuff. Isn't it? Oh, I see what I did. I forgot about that. Now I'm pointing to areolar and dense irregular. Okay. Yeah, this is the perichondrium. Yeah, just to see it again. We've seen this already. It's the lamina propria or submucosa, which is also areolar. Okay. So there you go. Another good example of that. Now, oh, this is neat. This is the trachea. But remember, back here is the esophagus, but I cut it out so I can have this area to put some text. But notice that the trachea is kind of shaped like a horseshoe. Kind of see like a C. You see that? But the back part of the C is enclosed in a little band of smooth muscle right there. You see that? That little band of smooth muscle is called the trachealis muscle. And the trachealis muscle is smooth muscle. So just to let you know, yeah, the back of the trachea is enclosed with a piece of smooth muscle. Just another good place to view smooth muscle. So we could zoom in on that and check it out. I think I did that. Maybe I didn't. But just to let you know, if you magnified on it, you could see that. But just, I guess you can't. But we've seen smooth muscle already. But that's what that is. I'm just letting you know. Now, the small intestine. All right. Nothing looks quite like this. You know, no tracheal rings, nothing like that. But you should recognize this is the small intestine. Obviously, you get these long, see these finger-like projections that are sticking on the inside? Only the intestine has that, okay? These are called villi, and the villi are in the intestine to help maximize the surface area. And you want to maximize the surface area for absorption, because this is where all your nutrients are going to be absorbed into the bloodstream. It's in these little villi. You're going to have capillaries and so on and so forth. But nothing looks like this. You know, if you got to have all kinds of cross-sectional views through hollow organs. You know, look at them side by side. The small intestine is the only one that looks like this. So this is the intestinal mucosa, or just mucosa here. The underlying submucosa, and then the outer muscular layer, or just muscularis, or smooth muscle layer. And most hollow organs are laid out just like this. You got mucosa, submucosa, muscle. You notice the three. There's going to be other tissues packed in there too, but these are the three main tissue layers. Get it? Mucosa, submucosa, mucosa, muscularis, or muscular layer. So we'll zoom in on that. So here we are, zoomed in. This is like 500 times looking at the intestinal mucosa. I just have mucosa in parentheses. And these are individual villi. See, coming up like this, these little finger-like things sticking up. And they're lined with simple columnar epithelium. If you look carefully, you can see that these are, remember I told you columnar cells have to have that vertical orientation? And you can tell that's one cell layer thick on these column-shaped cells. Say that real fast. The little white spots you see every once in a while, you see those things? Like holes. Those are goblet cells. Some slides, the goblet cells look like little grainy things, but here they kind of look like little empty pieces or something. Those are goblet cells. Anyway, simple columnar epithelium lining the villi or the mucosa. Here's a zoomed in even more. Well, I think this last slide, let me look back. This must have been about 100 times, and this one must be about 500 times, 400 times, something like that. Because here, it's a really good shot. You can see definitely, you know, that's one cell lit, one cell deep. See, there's no cells underneath it. The columnar cell, one columnar cell, that's a goblet cell. That's a goblet cell, right here. So, and then this is the other side of the of the villus. But it's just another good view of simple columnar, all right, in the mucosa. Here we're looking at the serous membrane and the capillaries. This is the outer wall. Remember serosa in my lecture. If you're not in my lecture, I apologize, but you're missing out. I talked about parietal peritoneum and visceral peritoneum, the peritonea or serous membranes. And so this would be visceral peritoneum covering the outside of the small intestine, which is a serous membrane or a serosa. And all serosae are made out of simple squamous epithelium, okay? And of course, capillaries, endothelium forming that too. I don't point out anything. I guess relying on stuff that's already pointed out. Let me go up. Wait, where am I at? Where am I at? Here, right here. Capillaries, submucosa, mucosa, muscularis. That's good. All right. Here's a really close up of, this is probably, again, 400 or 500 times looking at the mucous membrane, the serous membrane. And you can see this is the simple squamous. These are the nuclei of the simple squamous cells. And here's the cytoplasm. Remember I said that the simple squamous looks like fried eggs. You know, these are like fried eggs laid end to end. Here it says this image of simple squamous epithelium was taken from the outer surface of the small intestine and is therefore a visceral peritoneum or mesothelium. That's another word for it. This mesothelium will secrete a lubricating fluid, serous fluid, that allows the organ to move with frictionless ease. The tissue will also provide a uniquely smooth surface to reduce friction. Okay, so that's why you have serous membranes, to allow the organs to be nice and slick, you know. But it doesn't have to be very thick, you know, because there's not going to be a lot of trauma inside. So Mother Nature is very conservative when it comes to tissue. You're not going to lay down a big, you know, stratified area if you don't need it. You don't. Okay. Now, here's more of the small intestine, but this time we're looking at the mesentery that's connected to the intestine. The mesothelium, that's another word for the visceral peritoneum. Visceral peritoneum, mesothelium, same thing. And remember, it's always simple squamous epithelium. But remember the connection between visceral peritoneum and parietal peritoneum, the little liaison between the two, a little bridge, you know, that connects the two, that's mesentery. And so that's what you're looking at right here. This is a piece of mesentery on its way to connect to the body wall, okay? And you can see there's stuff in there, like blood vessels and nerves and so forth, and other tissue in there. And the underlying connective tissue, you're gonna have simple squamous on the outside, here and here, but the stuff that's on the inside is going to be areolar, and that's forming the lamina propria associated with the mesothelium. So areolar connective tissue forming the lamina propria of the serosa. So there you go. That's pretty cool. This is just a close-up of the intestinal mucosa, the underlying submucosa, which is dense irregular connective tissue and submucosa, and there's your smooth muscle layer, and then there's your serous membrane right there, okay? Again, just another view. The mesothelium, this is a close-up of the mesentery going up. This is the next to the intestine. But the superficial layer, because this is the outside right here, that's where you're going to find blood vessels. You get veins and arteries and capillaries, all right? How do you tell a vein from an artery? Well, generally you look at the thickness of the wall. See how thin this is? It just looks real thin, and see this kind of thicker. The thickness, again, is caused by the presence of smooth muscle. You can have a few smooth muscle cells and veins, but not nearly as much, particularly if they're side by side. You can tell them apart. Small lumen, thick wall, big lumen, thin wall. It's an artery versus vein. And of course, there's blood in there. The blood is vascular connective tissue. Name this tissue. Well, vascular connective tissue. How about that? Okay. Now, there's a lot going on in the small intestine, okay? Here's a close-up of the smooth muscle that's in it. Notice I have smooth muscle, circular and longitudinal layers. You have 2 layers of smooth muscle, and this is true for most of your hollow organs. You're going to have a circular layer that goes around in a circle like following the lumen, and then a longitudinal layer that runs the length of the intestine or whatever hollow organ it is, you know, the tubular thing. And these 2 layers of smooth muscles, they function differently. The circular smooth muscle decreases the diameter of the lumen. It squeezes it closed like that. The longitudinal layer shortens it. So you think about this shortening and squeezing, that's how peristalsis works. So that's how things get moved down the lumen, okay? Now, if you're taking a cross section through the organ here, the hollow organ, you can see this. And of course, this is a like a lateral view looking at in the sides of the smooth muscle cells. And this is beautiful because you can see the nuclei, the sides of the nuclei, and the long kind of stretch out nuclei, the long tapering cells, very characteristic of smooth muscle. But this, so this is now it's kind of confusing because this is circular smooth muscle, but a longitudinal section. You cut lengthwise. Now this is longitudinal smooth muscle, but cut in cross section. So you cut, these muscle cells are coming at you like this, like that. And so you cut them and you're looking into the ends of them. So here you're looking into the ends of the smooth muscle cells. Here you're looking into the sides of the smooth muscle cells because their orientation are like 180 degrees from one another. You see how that, this circular smooth muscle is going like this, longitudinal smooth muscle is going like that. And you should recognize in this smooth muscle, the large single nuclei per cells, kind of stretched out looking, but one big nucleus. In cross section, the cells are the nuclei are cut right in the middle. You see that? Very distinct. So there's your smooth muscle. Xiphoid. I forgot about this. It says absent on picture day. So we're not going to look at the xiphoid because I couldn't find any good pictures of it. So he missed out. So no xiphoid today. And so I think that's where we're stopping. I think we're going to stop. So it's going to be a little bit early today. Does anybody object to we finishing our PowerPoint 15 minutes early? And we'll do muscle types next week. And then after next week, you'll be getting ready for your practical. But I think this is, I'm saving this for next week. So there's muscle types. Let's see where we are. Muscle, yeah, there's not much left. Muscle types, nervous tissue, the bone, and then blood. And then that's it, la fin. So we're going to do that next week. And have any questions that you may have, or I don't know, maybe you have some questions right now. Let me look at you guys. I've got you minimized where I can't really see. But,
I actually have a question.
Okay, let me make this bigger so I can see you and hear you. All right. Okay. Somebody, I can't. Anyway,
what time do you want I through Z to come into lab? Like 11:20, or do you want us to come in?
Oh yeah, I forgot about that. I mean, there's we didn't go into lab last week, did we?
No, sir.
No, we didn't. And there's not really any need to go into lab today. I mean, there's nothing in there. There's really nothing in there to see that I haven't already shown you. Because what you need to study, there's nothing in lab that you really need to study. What you need to study is this PowerPoint, because this is where your test is going to come from. You're going to see all these pictures again, but I'm going to just ask you questions about it. Like today, for example, I pointed out these tissues, and for your practical, I'm going to show you this picture again. But I'm going to point this out. Say, okay, name this tissue right here. and you should recognize that it's adipose connective tissue and so forth. You see, so that's how it's going to be. I mean, if the lab, I said the lab's supposed to be optional. I mean, that was to allow this course to be hybrid, so to speak. But to me, it's I don't see a need to go to lab unless you really want to. I honestly, I haven't even been into lab all semester. But if you wanted to go in there, or we could use the extra time to answer other questions or whatever. Or if you don't want to do it in lab, we can Zoom. We can do it like during office hours, have a private Zoom meeting, and we can go over slides together, whatever you want to do. Okay, but to physically go to lab, you don't need to do that.
Okay, thank you.
Okay, anybody else? Yeah, we're good. So next week we'll finish this up, and then we'll get ready for y'all's first practical. It shouldn't be bad. Remember, your practical begins when class begins, and so your class begins at 10, right? And it's open just for 30 minutes. So from 10 to 10:30, 2 weeks from today. And that's it. And it'll be 25 questions, 30 minutes to finish 25 questions. And it'll be, 25 pictures with multiple choice questions. Just like it's all recognition. Okay. Identify this, identify that, something like that. So you good? No questions? Okay, well, give me a holler, email me or whatever if you have any questions between now and then. Until then, I will see you guys next week. All right.